them are easier to produce because you can prepare many tablets very easily. You can prepare them by extrusion, spheronization, without any need for processing them further. However, from our experience and from literature experience, a control on drug release is very difficult unless you use special type of materials, hydrophobic waxes, or high viscosity hydrophilic polymers. And we attempted, and I will show you later on, to use hydrophilic polymers, high viscosity hydrophilic polymers, but we were not successful in achieving controlled release with monolithic systems. While for core shell systems, we take our particles and then we apply an additional step of film coating, subsequent to active layering or extrusion or extrusion spheronization or mini tablet production. This is a slide that I use in my lectures, and probably you're all familiar with. This basically would show you that the multi-particulate system is superior in many aspects to a single unit system with respect to, for example, gastric emptying, inter and intra-subject variability, food effects, risk of dose dumping, and the ability to modulate the drug release. The only downside that you can think of is probably the issue of drug loading. They have a problem with drug loading, and with high-dose drugs we may have some problems. However, this can be solved using special technologies such as spheronization, for example, or mini tablet production, or as I'll show you in a minute, we have active layering for a high-dose drug, and we were successful in producing a controlled release system, an effective controlled release system. We attempted to contact the local manufacturers, the generic manufacturers, Jordan. We tried to explain to them why would they be interested in multi-particulate systems and why would they need to fund our work, contribute to our work. And the issues that we presented them with were as this, like this. Developing the multi-particulate dosage form is, in fact, a strategy that a big pharmaceutical manufacturer, an originator, may use in order to add a market barrier to generic manufacturers who do not have these technologies. So if you develop the skills, if you get the knowledge, you may be able to overcome this barrier and improve your chances in the market. Otherwise, you would have to buy these ready-made units from intermediate product manufacturers, and this will cut into your profit margin. And this is the case for our generic manufacturers in Jordan and in neighboring countries as well. In addition to that, with these multi-particulate systems, you may have the possibility or the potential to produce some innovative drug products of generic molecules. Modifying the pharmacokinetics, according to concentration time to file, may in fact improve the tolerability, reduce the dose that you add to your system or that you introduce to your patient, and probably minimize side effects. Probably you will enter into a pediatric market that is not addressed by the originator. So this is the rationale that we presented to the generic manufacturers in Jordan. And we got them to support us in a few projects. These are the projects that we've completed. The name of the students are here: Haytham Ahmad, Firas Faleh, Lima Al-Murra, Jalal Amir. Those are the students who participated in these projects as part of their MSc work. And we've addressed a group of issues that we developed with, or research questions that we've developed with the generic manufacturers. We, for example, with Haytham, went through the application of active layering to develop a multi-particulate system of a high-dose drug. We don't have the extrusion spheronization technology, so why not go for an active layering technology? Can we use it or not? Then we tried to play a little bit with the release rate from such systems. Can we modulate the release rate from such systems, for example, by modifying the configuration of the system, adding seal layers in different configurations? This was the work of Firas. Then we went into a more conventional approach, and we used a simple tableting technology, However, instead of a 10 millimeter tablet, we used multi-tip bunches with 2 millimeter tips, and we produced many tablets that are very similar to the beads or the pellets that are produced by extrusion sterilization, and then we coated them in a side-vented coating line, okay, conventional technology, in comparison to the first two projects where we used a fluid bed processing. Then, with Jalal, we went into a super generic point of view, or let's say an innovative drug delivery product, and we used this multi-particulate system in order to develop a fixed dose combination of two products. And the challenge was that we developed a fixed dose combination with dual drug release rate. For example, one of the components was a controlled release, the other was an immediate release, and this was done on the same particle. The release that was controlled and immediate was achieved from the same particle. So this would reduce the time of processing significantly. This was the project of Jalal that I'm presenting here. In the first project, we went on to evaluate diltiazem hydrochloride, a high-dose drug, highly water-soluble, and we tried to layer it onto saccharose pellets, the size of which was 500 to 600 micrometer, and we used polyvinyl alcohol, polyethylene glycol, graft copolymer, a water-soluble binder, in order to fix diltiazem on the surface of these pellets, and then we coated these pellets with a polyvinyl acetate coating, polycoat SR. And this was done in single buck process in uniglat fluidized bed processor, bottom spray processor. This is a schematic of the configuration of the system. The core surrounded by the polycoat IR with the diltiazem, and then coated with the polycoat SR, the polyvinyl acetate dispersion. Regardless of the target gain in terms of percentage, we got always excellent process yields, and we got immediate release for the first layer, and then we coated this layer with the polycoat SR layer to produce a controlled release system. We evaluate the drug release from these multi-particulates, and we got this figure. It's a very busy one, but the idea that I'm presenting here is that you can, in fact, get different release profiles using this technology. Modifying the weight gain of the coating material can produce a variety of release profiles according to your needs, to your product needs. Basically, this figure shows different weight gain of polyvinyl acetate, either plasticized or unplasticized, cured or uncured. And one of the messages that we got here that you have to plasticize polyvinyl acetate, despite what PSF were saying at the beginning that it's a polymer with a low blast transition temperature and you don't have to plasticize it, but we found that you need to plasticize it, and we shared it with them and they were appreciative of our observation. A very stable system. We put it on a stability study in closed HDPE bottles, and we got a release profile that was stable for a period up to 3 months. We didn't continue further because the student has to graduate, he has to leave, so we stopped the project at that point. But this was very interesting because these are systems that are liable to change in their performance upon storage, and they were stable in such a case. This is something that you can use in order to study the process more quantitatively and reduce the clutter. In the first graph that I showed you, basically these are the data, however they are transformed so that we can extract an apparent release rate constant and the mean dissolution time. And here you can see the apparent release rate constant decreasing with increasing the coating level with gain and the mean dissolution time increasing. And again, we have here the effect of plasticization and curing as well. This is scanning the electron micrograph of the surface of particles. The uncured non-plasticized particles. A beats the plasticized particles, and you can see it's a much smoother surface. And C is a cross-section of the bits, and you can see clearly the core, first layer and second layer, the drug-containing layer and the controlled release layer. Afterwards came another student, Firas. Firas was obsessed with the issue of fine-tuning, playing around with release rates. So the research question was that if we modify a little bit our system in terms of adding additional layers, modifying the configuration of these different layers, would we get different release profiles? We changed the drug here. We used caffeine, a moderately water-soluble drug, instead of a freely water-soluble drug. We changed the polymers here. We used Udrogets as both binding and controlled release coating materials. We used Udroget E, which is a polymer that is gastrosoluble as the binding polymer, and we used a combination of Udroget RS and Udroget RL as controlled release polymers with different configuration. The color scheme here is not that clear. My original presentation was nice green, red, and yellow, but I was talking to Dr. Riyad, changing the computer, changed a little bit the color scheme. Anyway, basically what we did here, we introduced a sealing layer, either above the drug-containing layer, below the drug-containing layer, or above and below the drug-containing layer, and we simply observed the release profiles. As you would expect, if you add a seal layer above the drug-containing layer, the drug release profile would decrease significantly. However, if you look at the blue squares and the yellow squares, these are configurations where we have a seal layer below the drug-containing layer, and they resulted also in some reduction in the release rate in comparison to the configuration, an identical configuration, but without this layer below the drug-containing layer. In fact, the mechanism, we're not sure of it. Maybe we have a two-dimensional or a two-way diffusion. Maybe it's because of the high hygroscopicity and water affinity of the polymer. We're not sure exactly. However, after that, we went to mini tablet technology. In the mini tablet technology, we prepared mini tablets that were 2 millimeter in diameter. We prepared them with methyl hydroxypropanethyl cellulose, high viscosity polymers, hydrophilic polymers. However, these mini tablets with the simple photogranulation technology, and coated them in coating band, these mini tablets did not produce a controlled release, simply because of the erosional component and the high surface area of the mini tablets. So we had to coat them with Udroget RS, and when we coated them with Udroget RS, and this is only part of the results, we got some controlled release out of these mini tablets. But without that, mini tablets as such cannot produce a controlled release, depending only on hydrophilic polymers. Now, for the fixed dose combination, we evaluated Kulikot IR and Kulikot SR, the same combination that I presented at the beginning, and we used a fluidized bed processor, and we attempted to develop a fixed dose combination of extended release metoprolol succinate and immediate release ramipril. The configuration was a core, then Kulikot IR plus metoprolol succinate, then a Kulikot SR coating, then a Kulikot IR with ramipril to produce the immediate release effect. Ramipril was released within 15 minutes, and this was a nice accomplishment considering the very poor solubility of ramipril. We have basically some sort of a solid solution or solid dispersion in this case for the ramipril. For the metoprolol, we found that the drug release was very much dependent on the formulation properties. We used, for example, 15% Kulikot SR unplasticized and compared it to Kulikot SR plasticized with the triethyl citrate, and We saw that plasticization reduced the release rate. However, we needed to add a seal layer in order to match the drug release of beta-log, the controlled release product of metoprolol succinate. One important point, beta-log here is a monolithic system, so a similarity of the solution in vitro does not necessarily reflect a similarity in vivo. However, it was used as a reference here for our formulation to, let's say, modify our formulation. Basically, we're successful in providing this dual-release multiparticulate system using layering and coating. Currently, I have 2 students who are working on 2 relevant projects, evaluation of the tablet stability and compatibility of these coated multiparticulates into disintegrating tablets, and the investigation into the preparation of a gas-retentive multiparticulate drug delivery system with also a fixed dose combination and a dual-release rate. I'd like to acknowledge the contribution of my former graduate students. Lima is currently in Hikma, Haytham is currently in Dardawa, Firas is currently with Jufar, Jalal is currently in Libya. I think he's still with the Libyan army because he was with Libya before he came to my lab. Current graduate students, collaborators, Dr. Ayman Abbas from Hikma, Dr. Nabil Darwaza, Dr. Ismail Khalifa from Dardawa, Dr. Shadi Madia from Hikma, and my collaborators from the University of Jordan, Dr. Khalid Aida and Dr. Bashar Khali. The contribution of the different companies is also acknowledged. PSF and Evonik have contributed significantly to these projects. Hikma and Dardawa have supported and sponsored the students. Thank you very much.
شكرا دكتور. المتحدث الثاني في هذه الجلسة زميلي الدكتور كارم الزعبي. دكتور كارم أستاذ مشارك ونائب عميد كلية الصيدلة في جامعة العلوم والتكنولوجيا، حاصل على درجة الدكتوراه في علم الأدوية السريري عام 2005 من جامعة هيوستن تكساس في الولايات المتحدة الأمريكية. دكتور كارم له أكثر من 150 بحثاً منشور في مجلات علمية ومحكمة. he is the guru in publication عندنا بالكلية بالأردن يعني. أشرف على أكثر من 50 رسالة ماجستير، وهو محكم علمي للعديد من المجلات العالمية والهيئات المحلية والدولية. حاصل على العديد من الجوائز العلمية المحلية والعالمية، كان آخرها جائزة عبد الحميد للباحثين العرب الشبان في قطاع العلوم الصيدلية العام 2012، وجائزة الحجاوي لأفضل بحث تطبيقي في مجال الصناعة الدوائية. اليوم التايتل تبع البرزنتيشن is the effect of Arbiotus and Racnin fruits extract on chronic sleep deprivation induced memory impairment, the role of oxidative stress. تفضل دكتور.
بالبداية شكرا for the nice introduction. This research that I'm going to present today is really the product of my collaboration with the pharmacology department in the university with Professor Firas Al-Ghani, who was the ex-dean of the college. The idea behind this series of projects was to kind of get the collaboration between these plant extracts that we have a lot of them in here in Jordan, and to try to find pharmacological and clinical uses for these plant extracts. Today's topic is the effect of Arbiotus and Racnin extract on improving learning and memory, cognitive impairment associated with sleep deprivation. I'm going to go through an introduction, aims of the study, the work, the method, and the results and discussion, and finally my conclusion. We are all fascinated with nature because nature is the major source for medications from the past eras, About 175 anti-cancer small molecules have been identified, so many anti-infective drugs, so many pharmacological properties has been identified for plant extracts and for plant-related materials. There is a wealth of appreciation today for the drugs, for the extracts, and for the active ingredients that comes from plant source. This interest is really coming back and going up these days. The pharmaceutical product market is really a big market today. Being in Jordan, Jordan is at cross-continent between Asia, Africa, and Europe. So it has a unique flora. And that's why we are in Jordan as pharmacologists and pharmacognosists are really have a target opportunity because we have a width of flora in Jordan that's a unique target opportunity really for us. The idea is, or the hypothesis that we're working on is the relation between neuroprotection and memory protection and the antioxidant properties of the plant. I have that hypothesis before working in plants that strong antioxidant chemicals are really good for protecting the neurons. However, if they come from a natural source, they will be more acceptable as neuroprotective agents. So that's not to treat an existing disease, but to protect the occurrence of the disease before it happens, like getting a certain pill, for example, for Alzheimer's, or for preventing the occurrence of Alzheimer's, preventing the normal consequences of the normal aging process, or the consequences of our stressful lifestyle. So that's basically the idea. So we went looking for plants that has strong or high antioxidant content. When there is a high generation of reactive oxygen species, neurons tends to go to apoptotic pathways and tend to die. So that's basically neurodegeneration. And that's where we start losing the neuron and we start seeing the negative effects on memory, on learning, and on multiple processes in the brain. Before working with Arprasis and Drachmi, we have also worked with Seratonia siliqua and with Hypericum perforatum, Hypericum tratifolium actually, and found that they are protective for psychosocial stress-induced memory impairment in animals. Of course, they're extracts. Arprasis and Drachmi أو بالعربي بنعرف احنا الجيجا، نبات الجيجا. استخدمنا the fruits of the giga plant, fruit extracts. This is the leaves of the plant. And it's, as a plant, it's rich with arbutin and monotropin. This is, as I said, is a collaborative research on the pharmacology part of it, so I'm going to concentrate on the biological activity of the plant. Now, when it comes to memory, memory comes in multiple forms. There are different types of memories, and each type of these memories has its own method for evaluation and for testing. Of course, in terms of time frame, memory is divided into short and long term, and it's divided into declarative and non-declarative memory. The type of memory that we are interested in here is the spatial type of memory. It's the kind of memory that you use when you want to find a location in the space, to find, for example, an address in a city. You know that it's in the middle of the city, so you can't come to it from the north or from the south. It's basically the memory that are mostly used by taxi drivers, really. As I said, you can divide the memory into short and long term. And when you say short, that's several hours, and long term, that's about 6 hours, going into days. Now, if you go over the days, if you go into months and years, then that's another form of memory that goes to a different brain area. So going into brain areas, the areas that are involved in cognition are the hippocampus and the amygdala, which are parts of the limbic system. Then you have the cerebral cortex. And on the lower part, beneath the limbic system, you have the cerebellum and the pons. So when the hippocampus is involved in the memory, that's really in the time frame of hours going into 1 day, going up into several days. When you go into weeks or years, it becomes a cortical memory. It's the function of the cerebral cortex. The hippocampus, as part of the limbic system, is involved in the spatial learning and memory, whereas the amygdala, as also part of the limbic system, is involved in fear and emotional memory. But all these memories and experiences will be filtered by the amygdala or the limbic system or even the cerebellum to be stored later in the center of judgment, which is the cortex. Now, sleep is a natural process. In sleep, the body in general gets the chance to revive itself, to relieve its oxidative stress, to do the normal, let's say, maintenance functions of the body. So a person without sleep is on high oxidative stress. If he doesn't sleep for several days, he might get psychiatric diseases, and that's been shown. So sleep is very important to maintain normal functions of the body. Now, when we analyze the sleep process, sleep is 4 stages. The last one of the stages is the REM sleep, which is the rapid eye movement sleep. That's the deep sleep. That's where a person really loses the muscle tone. That's where most of the dreams occur. And that's the most important part of the sleep. That's the part of the sleep that really gives you the feel of rest. So if a person loses the REM sleep, then he lost really the important part of the sleep, the deep sleep. And that's the important part of the sleep that's really important for normal physiological function. Now, it's been shown that many people are really lacking sleep. They do not sleep enough, or they do not sleep in a comfortable way that gets them really into the REM stage. We mimic that in animals, so we made them sleep deprived. So if we say sleep deprivation, that's a common problem. That's a problem for people who work night shifts, like physicians, like nurses, like guards, بكل المؤسسات الحكومية وإلى آخره. So sleep deprivation has been shown to lead to memory impairment and to lead to neurodegeneration. Now the issue is how to prevent that, or how to protect these people from the harmful effect of sleep deprivation. Our previous work in the lab has shown that, for example, supplementation of vitamin E can help in preventing the neurodegenerative consequences of sleep deprivation. Now we want to see or to find natural resources. So we decided to try the extract of apparatus and daphne. So the aim of the study was to test the effect of that extract, fruit extract, in its ability to prevent or improve memory impairment associated with sleep deprivation, and to find molecular targets. Is it really our hypothesis? Is it really oxidative stress? So in terms of experiments, we got the plant, the fruits, and we made the extraction process by standard methods that I'm not going into its details. We did also some fractionation. That's for the phytochemical analysis, not for the pharmacological study. The pharmacological study was simply based on the first extract of the fruit. So we had the following animal groups: the control, which was given the vehicle. Then you have the sleep deprivation, and that's basically animals that were sleep deprived, and I'm going to show the model in a minute. Then we have the white platform group, which is really a control for the sleep deprivation that I'm going to explain also. Then we tried the apparatus and daphne extract in 3 doses, 50, 200, and 500 milligram per kg. We tried it alone on normal animals, and we tried it with animals that are currently exposed to sleep deprivation. We tried a neuroprotection model. So in the same day we started the sleep deprivation, we also started the extracted treatment. And that's our idea, it's neuroprotection, it's not treatment. This is the sleep deprivation model. This is a diagram. This is a water bath, that's plexiglass, and it has in it 20 platforms. These platforms are small, 5 centimeters in diameter. So the animal, it's a rat, it's not a mice. So it's just going to sit on it, but he cannot, for example, sleep. And even if he falls into sleep, once he gets into REM, he will lose muscle tone and he will fall into water. So he will wake up and climb up that. So there is no way for these animals to sleep. Of course, this procedure is standard, internationally standard, and was approved by the Animal Research Committee and all ethical committees. So it is, and we did that for 8 hours a day for the animals for a month. So we did it during the day because these animals are nocturnal, so they sleep in the day and they wake up in the night. So we did it during their sleep time. We did it in the day, and that will make them wake up. So that's really a chronic sleep deprivation model. Of course, we tested the memory thereafter, after a month of treatment, 4 to 6 weeks actually. This is the radial arm water maze for testing memory. It really mimics the spatial memory. It mimics the taxi driver when he wants to find a place, or when any of us wants to find a place in a city. There is a hidden platform, for example, in one. It's underneath the water. When we do the testing, the room light is dim, so the animal cannot see. But he can remember. How could he remember? On the walls of the room, we have pictures, different pictures, of different shapes. The animal should remember depending on the pictures or on the visual spatial cues. So we give the animals 12 trials of training. That's just 12 consecutive trials of training. So we put him in any arm. These are arms, 1, 2, 3, 4, 5, 6. So you put the animal on any arm, and then let him wander around in the maze until he finds his way out. Of course, naturally, the rats do not like water, so they escape from water. Once he finds the platform, he will try to remember. We'll give him 15 seconds of wondering while he's on the plate, so he can look into visual cues. After the 12 trials of learning, we give him a trial of memory. That's the short-term memory. That's 30 minutes after the end of the training. Then we do the long-term memory test, which are done 5 and 24 hours after the training. The memory test is very simple. You just put him in any arm and find how many mistakes he will make until he gets it right. The mistake is the entering any arm that does not contain the platform. So it's really simple and based بالعربي متاهة. في بس متاهة بسيطة لتستنج الكوجنيشن of the animal. After the end of the month, the animals were decapitated using this machine, the مقصلة, mimicking المقصلة. And we did without anesthesia because that's how you do it for neurons work. You don't want the anesthesia to interfere with the neurons. We got the hippocampus and the cerebellum and the brain samples out of the animals, and we wanted to compare the important proteins levels and see if the drug really affected these proteins, the ones that are important for oxidative stress. So we measured the glutathione peroxidase, glutathione reductase, catalase, superoxide dismutase, and thiobarbituric acid reactive species. These are standard markers for oxidative stress. I'm going to skip the phytochemical because basically we have characterized the arbutin and the p-hydroxy acetophenone glucoside, which are known to be there in the plant. So this is the learning. This graph shows on the y-axis the number of errors made in the maze, and the x-axis is the learning trials, which are 12. And what you see here is the number of errors. And these are the groups. So we have the control, the sleep deprivation. Yeah, forget the white platform group. That's the control for the sleep deprivation. Remember that we put the animal on a 5 centimeter plate. We have a plate that's big, that's 12 centimeters, so he can sleep on it. That's a control for the environment. Because this is behavioral study, you have to control for the environment. Basically, in the learning, in all those, there was no difference between the group. Neither sleep deprivation impaired learning, nor process adapting in any dose extract, process adapting extract in any dose affected learning. This is the number of errors, and here is the 30 minutes memory test. That's when we tested them after 30 minutes. And as you see, sleep deprivation, and this is an application of previous studies found, sleep deprivation impaired short-term memory as indicated by the higher number of errors in the sleep deprivation group. Now for the extract actually reversed or prevented the impairment in sleep deprivation-induced memory impairment in 200 and 500 groups, while the 50 milligram group was not significantly different from the sleep deprivation. So we need at least to go to the middle dose for the protection. This is the 5 hours test. Similar results also were found. It's protective for long-term memory. And this is just a confirmation, the 24 hours test, similar results were found. Okay, now we have established that process and anxiety extract prevent sleep deprivation-induced memory impairment. Now is it related to oxidative stress? So that's where we started to look at the markers. This is the glutathione, which are the oxidative stress species. So if you have more glutathione, then you are more protected. We found no change. However, glutathione, when it's oxidized, it becomes glutathione. Sorry, when it's reduced, it becomes glutathione reduced, and that species was increased in sleep deprivation, and increasing that species indicates, okay, I don't want to take 3 minutes. Increasing that species indicates oxidative stress. So as we see, sleep deprivation has higher glutathione reduced, and apparatus and drafting prevented that. And the ratio of both is important. Again, it is the change in it due to sleep deprivation was prevented by the extract. This is the glutathione peroxidase, which these are the enzymes that are important for oxidative stress. So they indicate oxidative capacity. So sleep deprived animals has this oxidative capacity in their hippocampus. And that was prevented by treatment with the extract. Similar result, glutathione reductase was not different. Similar results were found with catalase and SOD, the superoxide dismutase, but not with the T-bars. With these results together, that indicates that there is oxidative stress due to sleep deprivation that was prevented by the extract. This is related to enzymatic capacity and related to glutathione pathway. It's not related to T-bars, which indicates the lipid peroxidation, which is a different pathway in oxidative stress mechanisms. This is BDNF, which is the brain-derived neurotrophic factor. It was not changed. This is a neurotrophic factor, it's an indicator of the neurons' function in the adulthood. It was not changed. So as a conclusion, 2 polyphenol compounds were separated and identified from the plant extract. Chronic sleep deprivation impaired short and long-term memory. It did not affect learning. The apparatus and drafting extract was neuroprotective at 200 and 500 doses, and even at 50 milligram doses, but the protection was limited because not all points were significant. It seems that this is through its antioxidative effect, because all oxidative mechanisms in the hippocampus were really reversed. The hippocampus is very small, حجم حبة العدس, يعني it's kind of specific. Not all the markers, however, were changed. but most of the markers were changed. So its oxidative stress is involved. We don't know if there are other mechanisms, but this is at least one mechanism. Thank you very much. And I'm ready for questions.
Thank you very much, Dr. Karim, for this very interesting lecture. Our third speaker of this afternoon is Dr. Iyad Rashid. He's a chemical engineer from the University of Birmingham. His baccalaureate, MS, and PhD from the same university, University of Birmingham, UK. And he's currently working as technical manager in JPM. The title of his presentation today is The Pharmaceutical Excipients of Tertiary Mixtures, a New Pharmaceutical Approach. Please, Doctor.
Thank you, Dr. Afifi. And thank you all for your attendance. The title of my talk today is going to be about pharmaceutical excipients of tertiary mixtures. Okay, it's just a title, but I have to stress on some introduction of what we did in our lab regarding binary mixtures. Because if you look at excipient development, you will find that, okay, it started with single excipients and then went up to binary mixtures, and they used different techniques. Nowadays, they are called co-processed excipients. So if you look at the top strategic development of new excipients, they are talking about co-processing of binary mixtures. Now, what we are handling currently in our lab is co-processing of tertiary mixtures or tertiary excipients. So basically, we have, before I talk about the tertiary, I have to stress on the binary that we did, because this is exactly what we're going to do with the tertiary. As an introduction, most excipients, they have different functionalities. Some excipients, they work as disintegrants, some they work as fillers, some work as binders. Now, this is the basic picture of each excipient. Now we looked at the overall picture. The overall picture talks about having all these functionalities in one single excipient. This is what co-processing, and this is what the excipient development is all about. Now, in application, some excipients, they work in wet granulation and others work in dry granulation. Now, the most interesting excipients, if you look in the literature, they are talking about the dry granulation technique. Now, it's easy and it's less expensive, less labor intensive than the wet granulation technique. So we stress on the dry granulation technique. Not forgetting that we have to have an excipient with multi-functionalities, with working as a binder and a disintegrant and a filler at the same time. Now, for example, if the most or the highly abundant excipient or the most common excipient in all pharmaceutical labs, like microcrystalline cellulose, the Avicil. The Avicil can be used in wet granulation and in dry granulation. And it has multifunctional purposes. But it has some kind of disadvantages. Its disintegration power is not enough. So if you, for example, if you have an excipient like Avicil with good disintegration power, then this is good. So based on this, we started processing our excipients, our binary mixtures, and we produced a couple of these excipients. The first one that we produced was based on the co-precipitation of silicon dioxide on chitosan. Chitosan is a highly abundant natural source. It's from crustaceans, from seaweed. So this is the first excipient that we produced. It's highly powerful excipient. This kind of excipient. It's more or less towards the microcrystalline cellulose. It has powerful binding capabilities and highly disintegration power. So, okay, it's satisfying. However, chitosan has got amine group. The amine group of chitosan is a bit interactive. It can interact with so many different APIs. So we stopped here and we thought of the origin of chitosan itself. The origin of chitosan itself is chitin. Chitosan is processed chitin. So we went to chitin and we did the precipitation or the co-processing of silicon dioxide on the chitin. Chitin at the same time is a bit less expensive than chitosan. I mean, the chitosan will cost around $35 to $40, while this will cost about maybe less than 15. So basically, we did have the same results. We did produce this kind of excipient. It can be used in wet granulation, dry granulation. It is powerful in terms of binding and disintegration mechanism. However, the co-processing with silicon dioxide is hectic and is a bit not scalable. So it's really hard to deal with silicon dioxide. Maybe well known to all of you as aerosol. Aerosol is, I mean, that big will weigh around 1 kilo. So it's really hard to deal with aerosol. It has very low bulk density. So we changed our direction into something called the metal silicates. Metal silicates, we looked at different metal silicates available. We found we have calcium silicate, we have magnesium silicate, aluminum silicate, and we did all the searching and all the co-processing with chitin. And with the chitin, we produced this powerful excipient which has high compression, high disintegration power and with good hardness tablet produced. At the same time, it's got some properties such as it has low humid uptake characteristics. Now, so we have calcium, we have calcium, we have aluminum, and we have magnesium. So what do we choose? So we did our intensive work on. Magnesium silicate. This was our focus. So magnesium silicate is different than talc. Talc, I think everyone here knows talc. What talc is? Talc is magnesium silicate, but it's natural. It's highly crystalline. So this magnesium silicate is different. It's synthetic and it's highly amorphous. So this is friendly to us. So we take this amorphous material and we exploit it further and further. So what we did is, do the same thing with magnesium silicate, go for the applications, and we found it's highly powerful when experienced on model drugs like ibuprofen and metronidazole, in terms of disintegration and in terms of tablet hardness capacities. Now, the most interesting feature that it's a work done in collaboration with Dr. Khulud Khamis. We examined the surface acidity of magnesium silicate and calcium silicate and aluminum silicate. We found out that the surface acidity or the surface pH of magnesium silicate is almost neutral compared to the basic calcium silicate and compared to the acidic aluminum silicate. So we have a neutral surface. Magnesium silicate's surface is neutral, doing this kind of experiment. Now, we examined the lubrication tendency. Now, most commonly, magnesium stearate is widely used as a lubricant. Now, magnesium stearate, stearate, not silicate. Magnesium stearate has a deleterious effect when added to so many excipients. For example, microcrystalline cellulose. If you compress microcrystalline cellulose, you get a high tablet with tablet with high hardness values. But if you add more than 1% or more than sometimes 0.5% and good mixing time, you will get the hardness of the tablets will reduce. Now, we discovered that having magnesium silicate lubrication does not affect it. So up to 10%, and that's the power of our excipient, of this kind of excipient, that it has the minimum deleterious effect of magnesium stearate upon lubrication with magnesium stearate. So this is a good starting point to us. So basically, if you add up to 10%, even for disintegration time, the disintegration time is not affected. Now, I talked about processing. Now, in our lab or in our company, we adopted a highly effective process, which is an alternative to wet granulation. This is the dry granulation. This is the core of dry granulation process or technique. This is called roll compaction. Now it's a technology which started to emerge recently. So we adopted this technique for our new excipients. We produced from this technology a new excipient which is based on starch compacted with magnesium silicate. Now, if you look at starch itself as an excipient is used as a filler up to 20%. It has been modified chemically, it has been modified physically to an excipient called starch 1500. Starch 1500 is a multifunctional excipient, can be used in direct compression formulation. Now, this is the level of starch 1500. It's a powerful excipient. Now, what we did, we started our research on starch itself, on maize starch, on the highly abundant source of maize starch. The source of maize starch is around 0.4 of a dollar. The cost of starch 1500 is 30, so it's a bit expensive. So we don't want to work on co-processed excipient. We still want to work on maize starch itself. So what we did, we just produced this kind of excipient by just compaction with magnesium silicate, and it's a very powerful excipient in terms of the properties that it shows and reflects when formulated with so many different model drugs. Now, how this happened? Some people may ask me how this happened. Starch, how roll compaction will produce or will turn starch, a non-compressible excipient, into a compressible excipient just by roll compaction, or how roll compaction will produce a brittle material like magnesium silicate into a highly compacted excipient with starch. So it has. I mean, if you look deeply into the mechanism of deformation, the type of excipients upon compaction will explain what things happen upon compaction. For example, if you look at starch itself is a highly elastic material. It's a plastic elastic material. Okay, microcrystalline cellulose is a plastic elastic material, but the elasticity of starch is high compared to microcrystalline cellulose. That's why it produces highly brittle tablets. Magnesium silicate is a brittle material, so we have it's a rule of thumb in the pharmaceutical industry. It's if you have a plastic material, if you have an elastic material combined with a brittle material, you will have a good excipient. A good excipient in terms of compression, in terms of compaction. This is all what we need. So we have this combination. Not only this. Starch, when starch is physically modified, let's say starch 1500, we have some slight gelatinization. Now, gelatinization of starch is a technique that they utilize in order to produce the starch 1500. Now, compaction of starch will somehow induce gelatinization. So you are basically, what's happening here is basically you have a gelatinized starch. So it's, okay, you started with a raw material starch, but during compaction, the high pressure, you have gelatinized starch. And here you go with this kind of excipient. Now, same goes with lactose. Okay, it's a different story, but it's another excipient that with Dr. Hatem al-Khatib. This is what we found. Okay, lactose is crystalline. Now, if you go to what they do with lactose and how they process it with using, like, for example, spray drying techniques, they turn the crystalline form of lactose into the amorphous form. Now, upon compaction, we have noticed that the lactose crystallinity is reduced, and in other words, the lactose amorphousness is emerged. So basically, you are changing the whole crystallographic structure of lactose itself. Now, if you have magnesium silicate with this kind of conversion, then you have a very good excipient. So basically, all what's been mentioned about before is all about binary mixtures. Now, where the tertiary mixtures are all about, where are they? So I leave this floor for you to think of. We have to think of co-processing using the same technique, using tertiary excipients. So this is the core of tertiary mixtures. We have to produce direct, we have to utilize we have to produce excipients for direct compression purposes with multifunctional excipient, and using this technique, rod compaction. We are utilizing this technology currently. And I will leave that floor for you to think of how so many different excipients you can utilize. And thank you all for this.
شكرا دكتور.
أنا ما حدا يروح لأنه في شهادات رح تتوزع.
لا بعد نخلص في سبيكر. المتحدث التالي هي الدكتورة لما حمادنة من جامعة الزيتونة. دكتورة لما تحمل درجة الدكتوراه من جامعة بوترا في ماليزيا في الموليكيولار ميديسن، ودرجة البكالوريوس من الجامعة الأردنية في الصيدلة. سوف تتحدث عن proteomics approach toward the identification of new markers and therapeutic targets for breast cancer. دكتورة تفضلي.
Quite different from the previous presentations, more to industrial. My work is more to basic sciences using proteomics, which is studying proteins profile of the cell to identify new drug targets and molecular or protein markers in breast cancer. We know cancer is a deadly disease. More than 7.6 million people died according to the WHO statistics in 2008. And among those cancer diseases, breast cancer is the most commonly diagnosed disease, and it's the most leading cause of death among women worldwide. In United States, in 2013, it's estimated more than 200,000 patients will be diagnosed with invasive ductal carcinoma, which is the most common tumor among breast cancer patients. According to our latest history, in 2010, around 980 new cases of breast cancer were identified, And among those, we are not including in situ patients. They just only include invasive carcinomas. In Malaysia, where the study was conducted in 2007, more than 3,000 breast cancer patients were diagnosed with that year. The most difficult part about breast cancer and breast diseases is that usually breast tissue is a heterogeneous tissue. It means if we want to identify a marker, a protein marker, a drug target, there are tumor cells in the ducts or in the lobular parts, and there are adjacent of heterogeneous types of tissue, adipose tissue by other types of cells that we cannot identify a proper marker for identification or a drug target in breast cancer. Metastasis in patients. And the treatment options, usually only chemotherapy would be given, endocrine radiotherapy surgeries would depend on the tumor size, lymph nodes involvement, and so on. Many of the patients treated for breast cancer, around 40% of them will be having relapses and they will die within a few years. So there is a need to identify breast cancer markers for early detection of the disease. The current detecting markers used are only estrogen receptors and progesterone receptors. They are used definitely to determine the patients who will take tamoxifen as a treatment. Also, HER2 receptor will be involved in patients also taking another type of treatment, which is Herceptin, for those patients only express this protein. We have a class of patients, they're called triple negative patients. They don't express either estrogen progesterone receptors or HER2 receptors. So there is a group of patients who cannot benefit from many of the medications that is available for breast cancer patients. The only molecular detected tool used is the BRCA1 BRCA2 mutation that it has a link in familial patients. So it can use to determine susceptible families of patients in susceptible families to develop in the future breast cancer. In this study, we wanted to approach the protein profile in the cells and to study if there is any link between what happens inside the entire cellular organization, what happens in the protein structure there in the disease state, and then to refer it out, compare it with the normal state of the patient. So we decided to apply one of the proteomics tools. At that time, when we started the project in 2005, two-dimensional gel electrophoresis was a very new technique in which we applied gel electrophoresis to study. In our study, we studied in normal tissues and the adjacent tumor cells. So the most difficult part in this project was obtaining fresh samples from patients undergoing surgery. We know that most of the surgeons, the main aim for them is to cut the tumor, put it in formaldehyde, send it to pathology lab, get identification. So for us, it was very difficult to convince them or to get through their routine surgeries in which to give us samples, put in liquid nitrogen, and give us two process for protein expression. So we lost many samples, and many samples were not used because we cannot have a proper protein profile obtained from the cells. Two-dimensional gel electrophoresis was coupled with mass spectrophotometer to identify proteins of interest or spots of interest, and then the proteins that we have, I will go to the discussion. The proteins, their protein profiles were compared between patients, tumor cell, and their adjacent node. And you can see, I know many of you might not be from proteomics or protein background. You can see that there is a difference in expression of different proteins among tumor and normal cells. We wanted to have our controls, so our controls were MCF7 and MDA in comparison with MCF10A. It's a normal breast tissue cell line. So we compared the cell lines with the normal cell line and later on I will show later on the tumor cells with their normal adjacent, from each patient. So from cell lines, we identified few proteins. Some of them were reported previously in many results. Some of them, like opioid binding cell adhesion, which was the first time to be reported in this study to be overexpressed in MCF7 and MDA, 231 cell lines compared to the normal cell lines.